Every cell nucleus in a particular living organism contains the exact same DNA molecules. Human DNA contains 3 million paired deoxyribonucleotide residues and contains 25,000 genes. The structure of DNA is understood of 3 levels of structure: primary, secondary, and tertiary. The primary structure of a nucleic acid refers to the sequence of its nucleotide residues. In DNA, secondary structure pertains to the helix formed by the interaction of 2 DNA strands. In the most commonly found form of DNA, 2 single strands lie side by side in an anti-parallel arrangement, with one running 5 to 3 and the other running 3 to 5. The 2 DNA strands are held to one another by base pairing hydrogen bonding between the bases attached to the sugar phosphate backbone. This base pairing is complementary. Adenine forms hydrogen bonds with thymine, and G forms hydrogen bonds with C. For nucleic acids, tertiary structure refers to the overall three-dimensional shape. In DNA, tertiary structure arises from supercoiling, which involves double helixes being twisted into tighter, more compact shapes. Example: plasmid DNA from bacteria. DNA is often called the blueprint for life because it contains all the information necessary for making the proteins required by living things. This information is carried in the primary structure of DNA. To pass information stored in DNA to a new generation of cells, DNA replication must take place. When DNA is replicated, each strand of the double helix serves as a template for the manufacture of a new strand of DNA. In each of the daughter DNA strands, one strand from the parent DNA is present. The production of new DNA is carried out by enzymes called DNA polymers. DNA polymers catalyze the addition of deoxyribonucleotide residues to a growing DNA strand. The parent molecule has 2 complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner A to T and C to G. The first step in replication is separation of the 2 DNA strands. Each parent cell now serves as a template that determines the order of the nucleotides along new complementary strands. The nucleotides are connected to form the sugar phosphate backbones of the new strands. Each DNA molecule consists of one parental strand and one new strand.